So first of all, I'd like to thank our sponsors. We appreciate your support for the event. And also like to thank our student volunteers who are at the back, and they will be collecting questions that you may have for the speakers. My name is Atul Varadachari, and I will serve as moderator for the discussion on the emerging era of personalized medicine. I will start off with a mea culpa. I will confess I'm not an IITian, and even worse, I'm not even an engineer. I went the medicine route, which is was the other major choice available to students in India at the time. However, I appreciate the organizers overlooking those errors and allowing me the opportunity to organize the life sciences thread for the conference. I have spent the last 20 years in life sciences and drug development, and I currently serve as managing partner of AlphaDev LLC, which is one of the leading developers of life sciences technologies in Houston. We work with academic innovators to create, manage, and fund early stage technologies. And some of our portfolio companies involve personalized medicine. So it is a special pleasure to moderate this panel. The evolution of medicine from the one-size-fits-all towards targeted and personalized medicine care is perhaps one of the great transformations in medicine in our generation. This ongoing shift is based on the convergence of a number of factors, including a better understanding of the biology of health and disease, tools to better analyze and interpret the expanding amount of information that is available, and new approaches to translate our insights into targeted therapies. Today, we are privileged to have 4 of the world's leading experts in these complementary fields who will help us understand the changes that are revolutionizing the way we understand and manage health and disease. So please reserve your questions for the end. We have our volunteers who will be distributing flashcards and collecting questions you may have and passing it up to the moderators. So to start, Dr. John Mendelsohn needs no introduction. He served as president of MD Anderson Cancer Center for almost 15 years, and under his leadership, Anderson grew to become the leading cancer center in the world on multiple dimensions. Since stepping down as president in 2011, Dr. Mendelsohn has served as the co-director of the Sheikh Khalifa Institute for Personalized Cancer Therapy at Anderson, which is one of the leading centers for this whole field. Dr. Mendelsohn has actually been a leader in what we now call personalized medicine since its inception. His early research in California involved the production of a monoclonal antibody, 225, that blocked the EGFR signaling pathways in cancer cells. This antibody went on to be approved by the FDA as a drug in 2004 as Erbitux, and it's now used for the treatment of colon cancer, head and neck cancer, and other areas. And more importantly, or equally importantly, his research really pioneered what is now the broadly adopted approach of anti-receptor therapy in the treatment of cancer. So there are few people in the world better positioned to help introduce us to the development of personalized medicine. Please join me in welcoming Dr. John Mendelsohn.
Good morning, and I'll give a short introduction to my topic, and then we're all going to welcome questions after we've spoken. This is a little over the 100th birthday of targeted therapy, which dates back to Paul Ehrlich. And on the right is his drawing, my poor copy of his drawing of postulated receptors on cells. You remember that if you look in a microscope at cells with staining, you see that some cells pick up one stain and some pick up another stain. That's how pathologists tell cells apart. And he postulated that cells had on their surface specific receptors or side chains for different dyes, for different dye molecules that were quite selective. And then on the right side of the slide, he postulated that if he had a bacteria with a specific receptor, he could attach a poison, that's the dagger, he used arsenic as his poison, to the molecule that bound to that receptor. And this was the first development of what became targeted therapy. His drug was called Salvarsan. He developed it in 10 years for probably less than $1 million. And we've come a long way, both in the expense and time it takes to develop drugs. Fast forward about nearly 80 years to the work that was just referred to by Atul. My rather crude drawing on the right here shows vertically a cell wall with a curve toward the right and a receptor in it called the EGF receptor, and then outside that cell wall a key called EGF. When you put your key in the lock in your car and turn it, there's an electric signal that goes out and lots of things happen. And if you pull the key out of the lock, the car stops. And that's the way the cells work in terms of whether they're going to divide or not. If a key, the proper key EGF, gets into the lock, the EGF receptor, a chemical signal goes out and tells the cell to divide. We developed an antibody shown in black there that would bind to the lock, that is the EGF receptor, and prevent the EGF from starting the cell from dividing. And it turned out that the EGF receptor is an oncogene. It's the product of an oncogene which can cause cancer. So we had been developing a drug that could prevent cancer cell growth by blocking a gene abnormality that causes cancer. And just for your interest, this is the first published evidence that blocking an oncogene product can inhibit the growth of a cancer cell. The open circles are, this is in an animal model. The open circles show the decreased growth of the cells when the animal was treated with our antibody against that receptor, and the filled in black circles were the controls. Now that was about 30 years ago, so we've come a long way. Now there are over 20 drugs like this that target various products of genes. So the paradigm we work on is that actually cancer is a disease caused by abnormal function of genes, that we have identified most of the genes that can do this. There are 22,000 genes in every cell in your body, and a few hundred of those control cell proliferation, those locks and keys. There are over 800 drugs in the pipeline, like the drug that we invented, the antibody that we invented, that target the products of these genes. So we're getting ready. We can now detect the aberrant genes in an individual patient's cancer. It took 10 years and $3 billion to sequence one genome. That's the genes in one person's body in the Human Gene Project. $3 billion, 10 years. Today we can do it in much less than a week for $5,000 or less, and it's going to get cheaper. So that means that we can take a tumor and ask which genes are abnormal in that particular tumor, and then we can pick therapy that is targeting the abnormalities in that particular patient's tumor, which may vary from one lung cancer to another, or from one breast cancer to another. Now, I'm not going to run through these examples in detail, but there are many examples of this kind of work resulting in successes. But let's look at number 2. Imatinib was developed to treat chronic leukemia because there is a gene abnormality in chronic leukemia that produces a product that imatinib can block. Now, doctors noted that this same drug can also attack another gene called c-KIT and its product that causes rare forms of stomach cancer. So here we have a drug that had been developed to treat leukemia that is now being tested, was now being tested against stomach cancer, not because of the tissue the cancer came from, but because of the gene abnormality in the tumor and the fact that a drug could attack that abnormality. And indeed, the drug works. So the same drug's now used to treat leukemia and this form of stomach cancer in a very targeted way. There are many examples of this kind of thing, and I want to assure you it's not that simple. It sounds simple, but most cancers, if they're going to be controlled in a long-term way, you need to attack 4 or 5 different genes, not just one gene. And occasionally, attacking one gene is satisfactory. But at least you understand, I think, the strategy we're using. Take a tumor, find out which genes are abnormal in that tumor. In my opinion, will be about a half dozen we'll have to attack in order to cure the tumor. If we can't cure it, we can at least prolong life by attacking 1 or 2 of those genes. And let's be specific and not give drugs to people where the drug won't work because that gene abnormality is not present in their particular cancer. So we created the Sheikh Khalifa Institute for Personalized Cancer Therapy at MD Anderson. We opened our doors about 1.5 years ago to, first we had to create the infrastructure to do all this work. The sequencing is sophisticated. The analysis of the data is even more sophisticated. The transmission of this information to the clinician and the patient is complicated. We had to design clinical trials that would bring patients and the drugs together. These are experimental drugs. They're not approved. You can't write a prescription unless you're doing a study. The FDA gets involved. It is a lot of paperwork. It's a lot of cost. We want to demonstrate the value of this approach, and we want to be able to eventually make it possible for every patient to have this kind of a personalized treatment plan for their cancer. Now, this is the first study we did. It's an important study because it was a proof of principle. This is a cell. There are, in yellow, are 4 different products of the cell that are like oncogenes. They're not all oncogenes, but they're important for controlling the cell proliferation. And there were experimental drugs available. And up until the time that we did this study, experimental drugs were given to people with advanced cancer. And the drug, there was no selection. Anybody who was dying of their cancer and nothing else was working were put on these new experimental drugs because the safety wasn't even sure, to work out the right dose and the safety. But we made the hypothesis that if we picked the experimental drug to use, maybe even for the first time in man, which was targeting one of those yellow objects on those cells, the patients would do better in these early trials with experimental drugs than if we gave just randomly these experimental drugs to whoever had advanced cancer. And indeed, that proved to be the case. Half the patients got random treatment, and the other half got treatment where the experimental drug was selected for one of those 4 abnormalities shown here, and those patients did better. And it was proof of principle that this approach would work. I'm going to skip that group of slides and say that we now have opened our doors. There are about 30,000 patients a year at Anderson who are failing standard therapy. We're following 100,000 patients. 2/3 of them are going to do well, but about 1/3 are going to need at some point experimental therapy or just stop treatment. And of course, we can't sequence the genes on 30,000 people right now. None of this is paid for. Most of this is not paid for by third party. So we now have the Clearinghouse Project, and this past 1.5 years, we screened 3,500 patients. It's the largest number of anywhere I know. This project is going on in a number of other great institutions too. And if we find something, going to the left on the slide, if we find an aberration in a gene that's treatable, we can put the patient on a drug if it's available, or put them in a trial if it's available. And if we don't, on the right side of the slide, we can study that patient's tumor and work with drug companies and try to develop new drugs that will target the product of that abnormal gene. So this is the results of our first 1,200 patients. And on the bottom there, you can see a list of the different genes. You can see it's not all 200 on our list. It's about 30 right now. And you can see that different tumors have different genes. If you work your way along, you'll see that the percent of tumors that had a gene abnormality, which we could target, is very low. So each cancer is a little bit different, which means it's going to be an interesting challenge. And we'll get more and more data on this. And I hope we'll have the arrows show where there is a drug available today that could attack the product of that gene. And of course, our goal is to assign a patient to get a hold of that drug. It's an experimental drug. It's hard to get experiments and patients linked together. And this shows you what the clinician faces. Those are all drugs in blue all over the place there. And the clinician has available drugs that attack many different steps. All these oblong circles here that run down toward the bottom, toward S6 and EL4FE. These are signaling pathways that can be abnormal in a cancer. So if someone, let's say there's a big circle in the middle, TSC2. Let's suppose we find a gene abnormality there. If we have a drug that can attack TSC2 or something below it, we could give it to the patient. It'd be silly to give a patient a drug that attacks something above TSC2 because TSC2 is already abnormal. It's driving the cancer, and that drug's not likely to work. That's the kind of thinking that's going on in our minds. So we have many challenges still. Tumors change. They're not the same all over even one patient's body. A lot of gene abnormalities don't drive the tumor. They're there, but they're not driving the tumor. We'd like to have better tests. We'd like to have blood tests that could pick up these abnormalities. That may happen. Tumor DNA is circulating in the blood of patients. We need more trials. We need to share the information that we have. We need to give better support to the clinicians and patients who don't know much about molecular genetics, but they have to make the decisions. These are very expensive. Most of these drugs cost $10,000 a month. So if they're approved, that's paid for by your insurance, and if not, it's quite expensive. And I guess the most important message is my last one. Half of all cancer deaths can be eliminated without any of what I've talked about if we change our lifestyle and our behavior, if we quit smoking, exercise, and get thin instead of fat. That will account for a reduction in 50% of all cancer, and that's personalized cancer medicine too. So thanks for your attention, and I'd love to have questions.
Thank you, Dr. Mendelsohn. Using the massive and exponentially growing amount of data generated by the scientific advances in the last 3 decades, including some of that extensive sequencing that Dr. Mendelsohn referred to, is both one of our biggest opportunities and, frankly, one of our biggest challenges in medicine today. Cerner is one of the leaders in this field, and our next speaker, Mr. Dick Flanigan, is well positioned to talk about the emergence and evolution of big data in healthcare and life sciences. Mr. Flanigan serves as Senior Vice President at Cerner, where he leads Cerner's premier focus organizations and is responsible for growing several of Cerner's largest and most strategic client relationships. He also oversees strategies and relationships with academic medical centers and pediatric hospitals and co-chairs Cerner's Academic Advisory Board and Pediatric Leadership Council. Through his 19-year career with Cerner, he has held a variety of general management roles, including Cerner's research and life business units, and has been instrumental in defining and delivering population health platform. Prior to Cerner, Mr. Flanigan spent more than 13 years at IBM. Mr. Flanigan has earned numerous management and leadership awards in his career. He serves on the boards of Donnelly College in Kansas City, Dynamic Clinical Systems, and the Tiger Institute. Please join me in welcoming Mr. Dick Flanigan.
Well, good morning, everybody. You did your mea culpa. I'm like the least credentialed guy on this panel. I'm the business technology guy, and here we're describing life sciences, and what an incredible group of professionals. And Dr. Mendelsohn, I have to tell you, for those of you that are, Houston residents or frankly, citizens of the world, MD Anderson is a global treasure. And sometimes we don't often take a step back and really take the time to understand what these men and women are doing at that institution that's impacting life around the world. And I have to share a story. We've had a relationship, sometimes rocky relationship with MD Anderson over the years. But I wanted to tell you, I met a nurse at MD Anderson some years ago, and it really gets down to personalized medicine. And we were walking through the lobby, and this was probably 1995, 96. And Debbie was a long time at that time. She'd been at MD Anderson for 35 years, and she said, people were taking their medical record in carts, and they were great stewards of their medical record because they were carrying it around with them, which was an incredible thing to see. And I turned to her and I said, Debbie, do you ever get depressed working here? I mean, I'm not a clinician. I've been in health IT for 20 years, and I said, is this hard to work here? She grabbed my arm and she turned and she said, 30 years ago, most of the people in this lobby would die. They came here because we were their last best chance. She said, today, half of these people will go home. And at some point in the future, most everybody in this lobby will survive. Now, that has had an impact for me in thinking about the role that science plays and the role that research plays in transforming care and transforming lives. And so a credit to the MD Anderson folks and many institutions like them around the world. The impact that we're seeing is really how Dr. Mendelsohn actually ended his comments, where there's lots that science can do, but frankly, there's lots that we as individuals can do. So I'm going to actually talk about a few different things, but I couldn't miss the opportunity in following an esteemed clinical leader to tell you a little bit about the impact. Because at the end of what we do in healthcare, there's a lot of value. You all work in other industries, and I will ask you, I'm going to ask one, actually, do any of you, and it's a little hard, how many of you work in a health or healthcare related field? Let me see any hands up there. Thank you. That's probably why you're here. And I want you to go out and talk to your colleagues that are not, because we need the help of all these really smart people represented at this conference. We need more people working on some of the challenges we face in this next generation of healthcare. Now, why is the healthcare big data thing? You all live with big data in all aspects of your life. You look at finance, you look at retail, you look at politics. Big data is pervasive in how decisions are made, how strategies are planned, and how those plans essentially get executed. But when it comes to healthcare, we've had these very troubling challenges of using big data to transform care. Why is it so difficult? Why is it that when you talk to most of your colleagues, they'll say, yeah, healthcare. There's an old Will Rogers joke, the great American humorist, and he said, when the world comes to an end, I want to be in Cincinnati. Why? Because everything there happens 10 years late. Well, that's health IT, okay? You have worked in maybe other parts of your career, and you've seen the promise of information technology being applied to very challenging problems. And yet, we are 10 years behind. Other industries have pioneered, so we're at the center of the oil and gas world. Now, there is a very interesting thing that's happening, whether it's social consciousness of folks wanting to change their career plan. We are starting to see in healthcare an infusion of talent from retail, from politics, from energy, from finance, actually coming to apply their skills. So one of my largest clients is an organization called Dignity Healthcare out on the West Coast, a faith-based organization. Dignity is building their warehouse, and you'd say, you're kidding, just now? Well, of course, they've had one, but it maybe wasn't that effective. And who do you think, what kind of people do you think they look for to hire to help build their warehouse? The traditional health IT guy? No. They hired a guy from Sears. Now you may say, well, maybe pick a different one, maybe pick a Walmart or whatever. The reality of it is other people have been figuring out the big data problem very effectively for a long time. And finally, we're now beginning to apply some of those skill sets to very, very large volumes of data. Now, you've probably heard some of these numbers. 90% of all the world's information has been created in the last 2 years. Does that seem odd to you? Well, if you use Snapchat, Instagram, I mean, think of all the stuff that's now permeating the lives of our children, ourselves, and tweeting and everything. We are moving massive amounts of information. Most of it, I would argue, is not going to cure cancer. But a whole lot of it tells us what's going on in people's lives. So 80% of all searches for healthcare start at a search engine. 80%. So there's a lot of ways you can predict flu in Houston. You can do what we did with the CDC, which is we looked why people come to the emergency room. We looked what tests were ordered and what the results of those tests were. That's an innovative way to do it. It's called Health Sentry, and it's worked pretty well. There's another way to do it. How many people are going online with upper respiratory distress or looking for cold or flu recommendations? Now, what an interesting use of big data to begin thinking differently about how we might want to apply technology to looking at both public health and, frankly, individual problems. So my first appeal is we need your help. If you're not working in some of this field, there's huge, huge opportunities. Now, within big data, healthcare has a very challenging problem that people at Sears and Walmart and BP and everybody else don't face. And that is, one, it's actually hard to get the full picture of the person or the patient. And that's because as an industry, we don't really have consolidated information platforms. We're working on it, but again, everything happens 10 years late. Secondly, we're not like the finance industry, which debit and a credit, and everything's denominated in dollars. We have a vocabulary, a semantic problem in healthcare. If I were to go to MD Anderson, or I were to go to Memorial Sloan Kettering, or Dana-Farber, or anywhere else, do you think they might describe everything the same way? I can assure you they do not. There's a lot more standardization than there used to be, and the cancer world, frankly, has been one of the best in creating common nomenclature. But maybe not great for 11 o'clock in the morning, but if you go on most nursing and you want to describe a patient's color, or you want to describe how they're feeling, it's like the art of medicine 101. How many different ways, how many different colors of urine are there? I know that sounds really funny, right? There are a lot. Now, I'm not sure how we reconcile those colors across the spectrum, but in reality, that's a problem not faced by lots of other industries. So, big data is here. It's finally being applied in a way that's beginning to impact the choices we're making, both for treating disease and also for treating individuals. So, at the end of all of the big data work is a person. And that's a little bit of the story for oncology. And there has been lots of focus on big data and disease, because big data seeks to do a couple things. What happened, what is happening now, what's likely to happen, and why did it happen? So we're being able to begin applying across lots of sites across the world these key points of learning. But in reality, what we really want is, we learn something, how does it work its way back into the clinical process? How does it inform care? How does it inform making better health decisions? And therein lies one of the challenges this industry still has not solved, which is, I learned something, how do I effectively change behavior of a prescriber, or as Dr. Mendelsohn said in his closing, behavior of individuals? Because as individuals, we influence most of the choices that will affect us throughout our lives. We say yes, we say no, we exercise, we don't, we do this. Genes play an important role, but we know they don't play the only role. So the opportunity that we're going to see in big data is companies like Cerner and Epic and Allscripts and Meditech and McKesson and Siemens and all the folks out there that are automating those core clinical processes. We're creating enormous stores of data. Increasingly, that data is being consolidated, reconciled, getting into a form that we can analyze. That is leading to certain insights that can then work their way back into those clinical processes and help us make decisions. The next major horizon for big data through outside these clinical processes is a field of research that is well recognized in other parts of industry, and it's comparative effectiveness research. What is the best, most cost-effective, leads to the best outcome of the health choices and the care choices we make? And the pioneering work there is a group up at Dartmouth and Dr. Elliot Fisher, and their work in saying, just because we can doesn't mean we should. And maybe some of the best choices involve doing nothing. And lots of work. So potentially in the questions, we'll get into a little bit. So we need your help. The field's expanding. We're getting our act together and getting the data consolidated. We're getting our act together and getting it normalized and semantically enabled and applying machine learning and all those technologies. But at the end of the day, we still have to ask these core questions. How do I get it back to the patient? How do I get it back to the prescriber? And then, did in fact I get the outcome that I saw that led the best quality, cost, and outcome? So with that, I'll turn it over to Atul and to the panelists and look forward to your questions. Thanks for the opportunity.
Thank you, Mr. Flanigan. It was very helpful. The previous two speakers have helped us appreciate our vastly improved understanding of not just disease, but normal health as well. But equally challenging is ways to use these new insights to provide a targeted therapies. And our last two speakers will help us understand some of the ways that we are bridging this gap. Dr. Jim Tour leads the Center for Nanoscale Science and Technology at Rice University, where he is the T.T. and W.F. Chow Professor of Chemistry, Professor of Computer Science, and Professor of Mechanical Engineering and Material Science. Rice University is in some ways the home of nanotechnology, the place where fullerenes were discovered in the labs of Dr. Smalley and Dr. Curl, who went on to get the Nobel Prize. And Dr. Tour has done much to advance our understanding of the field since then. Dr. Tour has over 500 research publications, over 60 patents, and has received many awards and recognition, including being named Scientist of the Year by R&D Magazine this year, and being ranked as one of the top 10 chemists in the world over the past decade. Dr. Tour's scientific research interests are extensive, spanning the gamut from nanoelectronics, graphene electronics, carbon nanovectors for medical applications, green carbon research in energy, chemical self-assembly, single molecule, nanomachines, and even K through 12 education. So he could literally be a speaker at any one of our conference's 5 threads. But we are delighted that he is with us this morning. So please join me in welcoming Dr. Jim Tour.
Thank you. So my task is to tell you something about nanotechnology and what's happening and how it might be applied in personalized medicine. Certainly, everything we do within medicine can be engulfed within the term nanotechnology. A protein is nano-sized and a cell is made up of nano-sized pieces. But more specifically, if we really think about where we might go with nanotechnology, think about, for example, bottom-up construction. If you think about how we are made, how are people constructed, we know from computer science that we cannot get complex structure by thermodynamic self-assembly alone. Thermodynamics will give us a pattern of A, or A, B. That will give us that sort of assembly. But if we want to have complex systems, we have to have non-regular assembly. And that has to be done by machines of some type. And what nature uses, it uses enzymes, it's its machines, so that when you eat a bagel this morning, it becomes a part of your ear by this afternoon because the molecules are broken down and the enzyme takes and assembles the molecules in some other location, sometimes modifying the molecular structure. And it does this, and it does it quite efficiently. And in some cases, those nanomachines might have ability to move across a certain gradient, and in other cases, they're pushed around just by the pumping of the heart, for example. Oxygen is carried throughout our body based on a heme structure, and those hemes are pushed around by the heart. But once they get to the location, there is recognition. So what might nanotechnology do? Well, if we have made, for example, nano cars and nano trucks. The idea is to be able to pick things up and move them from point A to point B. And these structures are only 2 nanometers by 3 nanometers. So I'll remind you how small these really are when they're 2 nanometers by 3 nanometers. That means they're about 10 water molecules across in size, which is really extremely small. Remember, if we have one swallow of water is 18 milliliters of water, which is 1 mole of water, which is 6 times 10 to the 23rd molecules of water in one swallow. If you had 6 times 10 to the 23rd sheets of paper, that stack of paper would reach from the Earth to the Sun 400 million times. So you have 400 million stacks of paper from the Earth to the Sun if you have a mole of paper. If you have a mole of water, you swallow it in one swallow. When you have entities that are this small, can we program them to do assembly from the bottom up? Can we give voltage pulses and cause them to pick something up and move it from point A to point B? So that in 100 years or 200 years, might we be able, for example, to build a building by just bringing in nano-sized entities, bringing in basic molecular structures and letting them build the building. And that sounds really far-fetched, but if you think about it, that's how you and I have been made. Everything in nature that is of complex assembly has been made from the bottom up. There is some thermodynamic assembly, but the complex assembly is all made by nanomachines, nature's nanomachines being enzymes. So what we're developing now are what we call unimolecular submersible nanomachines. And the idea is to have them be able to go and recognize a cell surface, and then we turn them on. And we can get little motors within these 2 nanometer by 3 nanometer machines to rotate at 3 megahertz. That's 3 million rotations per second. And can we use these to momentarily open up a cell membrane to allow things to diffuse in? This is where nanotechnology might take us. Well, where are we today with nanotechnology for medicine? Well, there are things that nanostructures can do that go beyond merely delivering. So the idea with nanotechnology first was to use them as carriers, and in fact, Dr. Ferrari was one of the founders of thinking of it in this way, of using a nanovector to deliver material to a site of interest, where you would take a nanometer-sized particle that might carry thousands of small molecules with it. You deliver it to the site of interest using, for example, a monoclonal antibody as things that have been developed by Dr. Mendelsohn to a particular site. Now you have a high local concentration of the molecule of interest all at that site. But can you do with nano something that a small molecule or an enzyme could not do? And what we're finding is the answer is yes. We're finding that we can take nanoparticles of a particular type and, for example, convert superoxide to oxygen, to O2, directly, not going through hydrogen peroxide as superoxide dismutase might do. But in any sort of trauma, any sort of traumatized material, there is overexpression of superoxide that can be deleterious to the tissue. And what we're finding is we can take nanoparticles of a particular type, of a particular design that have been functionalized to increase their half-life in biological organisms, and then convert that superoxide directly to oxygen in large amounts catalytically. So you take something and you take a ischemic material and you give it exactly what it needs, oxygen, in that place. And this is something that a small molecule and an enzyme cannot do. So what we're finding in this nano world are things that cannot be done with small molecule and enzyme, and this is being done today. In fact, we have a lot of discussions all the time with Atul about the efficacy of these compounds and what they might mean. But remember, the target for the future is to do bottom-up construction. You say, well, can you really build quickly with this? Well, some strains of grass grow 2 feet tall in a single day, which is pretty fast construction. And the complexity within that blade of grass is more than the technological complexity within this room and within this building. And if we increase that by an order of magnitude, we'd get 20 feet tall in a single day. So you can build from the bottom up, and that's where nanotechnology will take us. Thank you.
Thank you, Dr. Tour. That was great. We now pass to one of the most challenging aspects in personalized medicine, which is translating all of these fundamental discoveries from the bench to the bedside, including designing drugs to intended targets. Dr. Mauro Ferrari serves as president and CEO of Houston Methodist Research Institute, where he holds the Ernest Cockrell Jr. Distinguished Endowed Chair. He is also executive vice president of Houston Methodist Hospital System and the director of the Houston Methodist Institute for Academic Medicine. Dr. Ferrari is one of the founders of biomedical nanomicrotechnology, especially in their applications to drug delivery, cell transplantation, implantable bioreactors, and other innovative therapeutic modalities. He has published more than 250 scientific articles and 6 books, and is the inventor on more than 30 issued patents and many more that are pending. He has received many honors and research funding from NCI, NIH, Department of Defense, NASA, NSF, DARPA, I can go on and on. Dr. Ferrari is an academic entrepreneur par excellence. Apart from his very impressive academic career in leadership, he has also created several companies that originated from research in his laboratory. So he is the perfect person to help us understand how research in personalized medicine is actually being brought to patients. Please join me in welcoming Dr. Ferrari.
Thank you, Atul, for the kind introduction, for the invitation. Thanks to all of you for being here. And I am a great fan and supporter of nanotechnology. And I would never be able to tell you why with this anywhere near the eloquence that Dr. Tour has just shown to you. I'm a great supporter of the notion of molecularly targeted therapeutics in cancer and the molecular genetics that is based on in cancer genomics. And I would not be able to come anywhere near the elegance of expression and the persuasion that has transpired from Dr. Mendelsohn's work. So what I'm going to tell you about is some doubts. I'm going to tell you about some of the things that, the pangs that I feel sometimes in my conscience when I'm looking at the future, how we're going to be attacking cancer and frankly many other diseases because of the same reasons. And as I'm looking at where we are, I am hoping to the good Lord, the smarter people than I am, can figure out a cadre, a set of genes that really make the whole difference for what pertains to the development of cancer and so that we can treat them in a targeted fashion. I'm a big supporter of that notion. I hope that it is half a dozen, perhaps 20, perhaps 3. I don't know what that number is. I don't have the basis for making a statement of that number. But the things that bother me a little bit deep inside is that, but many of the patients many of the people that actually carry some of those mutations that have been identified never develop cancer, as far as we know. Or perhaps they do develop those cancers, and there are defensive mechanisms, protective mechanisms in the body that kill those little cancers when they develop. Perhaps this happens many times during our lives, and we never know about it either. So perhaps there is a whole different set of genes that deals with actually stopping the progression of disease or maybe keeping it in check in ways that then allows other things that happen in life to take care of them. So perhaps the set is larger, and as we know, the cancer is a disease of genetic instability, and the same individual cancer inside of a person changes over time. You get different metastases from the same primary disease, even if they're in the same organ. Over time, their genetic mutations evolve over time, and so that gives a set of targets that are moving. So that makes life a lot harder. But assume for a moment, as I think will be possible one day, assume for a moment that we get to know all of those cancer genes that are troubling the lives of so many. Then we are going to be able to attack them and cure them. The question is, we have known about genetic diseases that are much simpler than cancer, right? They go back to one mutation in one gene. And we have known about those for a long, long time. And how many of those have we cured? Essentially none. Why? Not because of a lack of molecules or for a lack of biological understanding, but for a lack of delivery mechanisms. We may have the solution, we have the letter with the magic message that solves it, but we don't have a way to get that letter to all of the residences, to all of the cells, in a way that takes care of the disease. It appears from the outset to be the problem of writing the letter and having the postman that does the delivery job. It would appear like the two things are separated. It's task A and task B, let's find it together. I'll give you the letter and we'll teach you how to go to the right places. It is not as simple as that. As a matter of fact, as you're looking at what cancer is, there is an emerging line of thought that looks at cancer as a disease of dysregulation of mass transport at the molecular level, at the cellular level, locally in a distant fashion, local tissue, different ways. So perhaps those very same difficulties in getting things to the right place are in reality the very essence, in many ways, of many aspects of what cancer is. which speaks for greater integration of the delivery sciences, so which nanotechnology is one, but is not enough, or which nanotechnology is certainly one. And the biological sciences, the molecular sciences, and the clinical disciplines that drive us toward the delivery of treatment to patients. So we talk about transport oncophysics. The physics of transport in cancer are really different from the aspects of mass transport in physics in health. And that certainly has a basis of genetic mutation someplace. Everything does. Everything in biology has got a genetic basis, right? So there's got to be an intersecting point someplace. But it is very difficult to get collaborating programs that look at that integrated perspective, and I think that really stands in the way of making a lot of progress. I will tell you a little story. And again, the only reason why I'm saying this is not to criticize the nanos for their delivery problems or to criticize the folks that work on the molecular genetics approach. These are pieces of knowledge that are absolutely fundamental and necessary. What I see that is not happening as much as it should is the integration of the two. So I'll tell you a little story to conclude my brief remarks here. And it has to do with perhaps the most exquisite molecular therapeutics targeting class ever. It is the notion of siRNA therapeutics. Of course, we've all been around, presumably, through the great promises of gene therapy that never happened, of oligotherapy, of anti-sense therapy. Let's talk for a moment about siRNA therapeutics. Since the day in 2005 when the Nobel Prize was won for the discovery of these classes of molecules that are really important in determining what from the blueprint that is in the genes is actually manufactured, the proteins then come out of that actually make things happen in the body. So siRNA therapeutics, the notion of being able to interfere with the genes selectively so that they cannot express the proteins that they actually that they encode. So since the discovery, 2005, and Nobel Prizes, pretty much many different companies have invested a huge amount of resources in delivering siRNA therapeutics. Pretty much every laboratory in the nation, perhaps in the world, that works with cell biology, uses siRNA molecules to modulate the behavior of cells in a petri dish in the laboratory. But then you try to do the same, you can silence an oncogene in your cells in the laboratory, very easy, but then you put that in an animal, you put that in people, those molecules get destroyed immediately by the body's protective system, by the certain classes of enzymes. So what's the story? Well, Roche Pharmaceuticals put in $700 million to create a line of drugs based on siRNA therapeutics, got zero. Merck put in $2 billion into that. No drug available yet. So at a certain point, a little drug delivery company went up to Roche, and essentially a company that was worth at the time $20–30 million on the stock exchange, went up to Roche and bought the whole thing, the $700 million worth of their operations, for an undisclosed price. And you would think that perhaps as a change in paradigm is a change of power structure. At one point is not a line. Second story, last Thursday, got a call from Merck that said, remember those $2 billion that we put into delivery, $2 billion plus, it was $2 billion some times ago. And said, oh, look, we would like to sell it to you guys for $100 million. Will you take it? I'm thinking, what? 2 points now, if I remember geometry, I'm a mathematician by training, 2 points actually do make a line on in a flat world at least. So I'm thinking, but is that the collaboration that I was thinking about? No, it isn't. No, it isn't. It is still two separated worlds that somehow are trying to eat each other up or take advantage of each other. This is not what we need. Ladies and gentlemen, what we need is a breakdown of these railroad tracks that we have laid for the way we are going to be developing our individual fields of knowledge, and is an argument for figuring out ways to work together. It's going to be nano, it's going to be molecular, it's going to be micro, it's going to be this and that and the other. It's going to be a bunch of other things that we don't even know yet. But to me, the greatest commitment to developing personalizing medicine is breaking down those walls, rolling up our sleeves, and figuring out a way to work together. Thank you very much.
So let me start off with a question for Dr. Mendelsohn. Thank you. So, what you're doing at the Institute is wonderful, and you've sequenced the genomes or done deep sequencing the tumors for a large number of patients. Is your thinking now that every cancer patient should get that kind of sequencing done? And if so, what do we do with that data?
We're pioneering something that's not, I think, necessary for every cancer patient now. Probably close to 2/3 of cancer patients don't need this. Standard therapy is either going to cure them or let them live many years. But for patients where standard therapy has failed, and that's 1/3, which is still a whole lot of individuals, this is something that can sometimes point them toward an experimental therapy or a new approved therapy that might help their cancer. And therefore, as the price has come down and the informatics and explanations for clinicians and patients have improved, I think in the next few years this will become a standard approach. We don't need to sequence the whole genome, though. There are only right now about 24 drugs, so we need to sequence points of mutation in a few genes. Probably 50 genes would be plenty right now, and we can add more as we learn more. We don't need to put in the expense of sequencing the whole genome. But we get patients every day that come in with a disc that they paid for to get their whole genome sequenced, and they hand it to us and say, what can you do with this, doctor? And that's a big challenge.
Well, as a follow-up to that question, at least today, is it only at the academic medical centers that you can do something with that information? And when is it likely to percolate out to the community?
No, you can use it in the community too. We share the information, and community doctors are ordering these tests just as frequently as doctors in academia. 80 to 90% of cancer care goes on in the community, and we share the information, as Dr. Ferrari pointed out. That's very important. We share it quickly, and there are a number of businesses now that make this available to community doctors.
Thank you. Moving to Mr. Flanigan, is big data still something we see primarily in the research and analytics world? How much is it starting to move to the real world, translational medicine and healthy? Are there examples you can amplify?
Yeah, there's clearly very good work being done in a lot of major research centers around big data. There's the core informatics work to reconcile the data, get it normalized and searchable and analyzable, and that's still a tremendous amount of work to be done. But there's applications that exist today. I'll pick one that I think again, fits in the personalized category, and that's sepsis. Leading cause of death in hospital is sepsis. Now, these are very sick patients. Some come in very sick, some get sicker when they're in the hospital. Being able to identify how to treat those patients and to identify that condition quickly is probably the biggest predictor of the outcome. And it's a problem throughout both rural America and in America's largest cities and biggest medical centers. So the data's been out there, the research has been out there for a long time about how to recognize and how to treat sepsis. And yet, reconciling that into a plan of care that creates a standardized approach across multiple sites that we can then analyze. Did this work? Did that work? Did it lead to saving lives? So one example that we've seen that went very quickly from big data was something that a large organization, the Catholic Health East Trinity, which is a big organization, Detroit, now on the East Coast, and they wanted to tackle sepsis. They did the research, they gathered the data, they proposed an approach that kind of ran in the background, and the punchline is using these big data approaches going right back down to the clinical process automation. 500 lives were saved. Now, that's real impact, and it's frankly, it's not genomic, it's not nano, all very important things. There's blocking and tackling work to do in healthcare today that benefits from looking at literature, analyzing, applying, implementing, and tracking. And sepsis is probably one of the best examples out there. There's been great progress throughout hospitals across America, but we're still talking about the leading killer of people, leading cause of death in hospitals today.
So I think that's an example where big data has made an impact, and there are many, many examples like that to come.
Thank you. So, one of the questions is, flipping it from the kind of example you gave to, as a layperson, as a consumer, what can we do to access or use big data, and what venues are accessible?
Well, as a consumer, I mean, there's a very high profile one right now called 23andMe, which many members of my family, I got everybody's 23andMe profile a few years ago, kind of what I call recreational genetics. And it's been kind of fun. We haven't really taken it that seriously as a family, but that's actually one of those big data initiatives that's actually come under, I'm sure there's folks on the panel that have feelings about that, but that's clearly, it's under FDA review right now, so whether or not that's the right way to do it. But there are those kinds of, I call them recreational examples. Really for consumers, I would say we're seeing big data around whether it's how we're comparing hospitals, comparing physicians, which is a very imperfect science today. But I mentioned in the tail end of my remarks around this emergent field of comparative effectiveness research. And I think that's really where consumers will begin to see the impact of big data, because it helps you and I make choices about who best can serve our family, who would lead to an outcome that we can afford, that's accessible to us. And I think the federal government's done some good work, and there's a lot of money that's gone into this research area and this area of innovation. And I think that's probably the place that will become very accessible to us, is in our making informed healthcare decisions.
Thank you. Dr. Tour, one thing that we hear about often is safety and toxicity concerns from nanotech, both for the individual and for the environment. Why do these concerns keep coming up? Are they being adequately addressed? Are they reasonable?
Yes, it's always reasonable to have safety and toxicity concerns. Probably as a field, there's nothing that's been there's no field that I know of that has been addressed from the beginning with so much care as in nanotechnology as far as the toxicity issues are concerned. The problem comes if we take, for example, a nanoparticle and we find out that it can go through the skin and it can penetrate the cells and it's toxic, to not think that all nanoparticles do this. What we've found now is that it's not just the size that controls this, it's the surface charge, it's the molecules expressed on the surface, it's the shape of the nanomaterial. So as that varies, all of the toxicity profiles vary. Now within the field of organic chemistry, I'm a trained organic chemist, we know that if you change the orientation of just a single methyl group, for example, on a steroid, you cause it from being a drug that can save a life to a drug that will rapidly kill a life. So what we do within organic chemistry is we look at each compound in and of itself, and we do a toxicity profile and a bioavailability profile on each entity by itself, realizing that any small change can bring a new change in properties. So as we're doing this now with nanotechnology, we see that there are some compounds that can be very dangerous and some compounds that are innocuous. But it's something that we have to do right from the beginning of the research to begin to, initially when we make something, assume that it's toxic and work with it in that way, and then bring it through a number of toxicity profiles, just as we would any new organic compound, and do an analysis in that way.
Thank you. You alluded to the fact that pretty much everything in health, humans, medicine in some ways is nano. And that word has been used extensively, one could even argue indiscriminately. So it's not quite clear what we mean when we say nano. Do you have a preferred definition or framework when we think about nanomedicine?
Well, the National Science Foundation says that nano is if you have an entity where any one of its dimensions is less than 100 nanometers, they consider that nano. Now that will encompass almost everything in biology below the cellular level that you can find it. So that was a broad definition. Now I remember when they were trying to define what it was. And in fact, I gave them a different definition, and I said nano could be anything where researcher wants to get money, they might call it nano. So it depends on what you want to use as a definition. But within the world of nanotechnology, I think things are moving to an extent where we can have sophisticated nanosystems that recognize that are what we would call active nano, in the sense that a photon of light comes in and an electron shoots out, versus a passive nanosystem where just its presence is smaller than a certain dimension, whatever we want to assign as that dimension. But yes, it is a very engulfing sort of term, and I'll admit that. It's very much like chemistry. Chemistry is extremely broad. You could do the same with chemistry and say that everyone and everything in this room is chemistry. And this is something I try to convince my children of. This is a very important field that I'm involved in. It's all encompassing as a chemist. So nanotechnology might not be as broad as chemistry, but it is a broad term.
Thank you. Dr. Ferrari, you alluded to the fact that if we're going to get to something from a personalized medicine perspective, you have all of these different fields that have to come together. How does this fit within the broader regulatory framework? I mean, is the way regulatory framework works today something that will accommodate our ability to develop personalized medicine?
Well, thanks for asking that question, very important. Of course, the perspective of nano meets pharmaceutical world is that you can think about personalizing the vector, not only the drug, but also the vector, because of course transport mechanisms are different in different people, and in the evolution of their diseases as well. So that is how they come together. Of course, the notion of the regulatory framework is a tremendously important one. And I would venture to say that the traditional regulatory framework of the Food and Drug Administration and its counterparts around the world is not very suited for personalizing medicine because of the sizes, of the ways in which clinical trials are run, necessarily with a very large number of patients, and also the way the pharmaceutical companies have evolved to focus on those drugs that can be sold to many people, cookie-cutter type of approach. And there are some arguments that one can make, and you've probably heard them all, in favor of the fact that you can evolve the regulatory framework to some extent, that you can redesign the strategy for the blockbusters along the different pathways, fundamental importance of cancer as opposed to the tissue of origin. So, I mean, there are certainly answers that the community is working on developing. But by and large, it seems to me the progress in regulatory approval framework processes have been very slow. It seems to me, in particular for what pertains to nanomedicine, that we were better off when we started this initiative. It was the National Cancer Institute when it started. The Cancer Nanotechnology Initiative I actually put it together. And back at the time, under critical path initiatives by the Food and Drug Administration, we were having what I thought were very insightful and collaborative conversations. And some of the drugs that we now have in the clinic right now, there are about a dozen different drugs that are nano formulations of drugs. And they're used as, well, some of them actually have reached blockbuster status. So back in those days, it seems to me that the level of the conversation was a lot more intense and insightful than it is right now. It seems to me that frankly, don't tell anybody I said it, but it seems to me that the regulatory body has been in a process of involution, not of evolution, and frankly, the regulatory strategies in other parts of the world, especially Europe, have leapfrogged us in terms of the way, their ability to integrate innovative perspective. But the short answer to your question is yes, we need different perspectives for personalized medicine. We need to personalize the FDA.
Well, the other comment, one of the other comments you made is really this field in some ways has been cyclical, right? So you had a lot of excitement earlier on, we hit some roadblocks, again there was some excitement. So it seems like, we kind of are going in waves. So do you have a sense for why that is and which part of that wave we currently are in right now?
Well, thank you. There are a couple of components to the story. The first is the reaction of science to a new domain discovery perspective. The typical reaction of the scientist is the generic scientist, if you will. is the first time you see it, you say, that's impossible. Then you show it's possible, and then the answer is, well, it's possible, but it's not important. Then when you show that it's also important, then it is, oh yes, I always thought I was the way to do it, and I did it first 30 years ago. So that is kind of a little bit of the cycle is explained in terms of the scientific response to innovation. But if you look at it in terms of translation to the clinic, I think pretty much all of the great discoveries that I can think of, or the great new approaches that I can think of, including monoclonal antibodies, perhaps that's a great paradigm to look at, and Dr. Mendelsohn certainly is the master of masters. There was an initial moment of great excitement, which got caught on by the scientists and by the pharmaceutical companies alike, by investors community. Then, when reality sinks in, and you have to see how many difficulties you have to overcome for a field to become mature, then there is the opposite, the pendulum swings the other way. That has happened, I think, in all fields, certainly happened in nanotechnology. But then if you look at where the pharmaceutical world is right now, monoclonals are clearly the triumphant class of molecules. So it takes time, it is cyclical, it has to do with the dynamics of human psychology and the dynamics of the market and cycles of investment. It seems to me that nano started out with great excitement, and then there was, the first nano drugs actually were approved close to 20 years ago. Before the word nano was really used, ribosomal formulations meet the definition of nano any which way you want to have it. But following that initial set of successes, there's been a lot of doubt, and now, as I was mentioning, the companies that are looking at the delivering, for instance, siRNA therapeutics, there are 3 of those. They're billion-dollar companies, and they're growing very rapidly. So it seems to me that the swing is back favorable to that discipline. But I'm not a person, I really don't want to be categorized as a supporter of nanotechnology. I'm a supporter of doing, like Dr. Mendelsohn says, I'm a supporter of doing whatever it takes to cure people. And if nano's got a part, however nano that may be, good. But it certainly is not going to be the holy grail of salvation. It is going to take integration of multiple different fields, and anybody that fixes or one set of tools is doing two things.
Thank you. So I have a couple of questions which I'm going to integrate, which is this broader role of personalized medicine. I mean, obviously things, John, you alluded to reduction of smoking, getting healthier. Those are not very expensive. In fact, you could argue you save money by not smoking. But outside that, a lot of personalized medicine is expensive. So the question is, what's really the price curve? What is the impact of this in potentially making a difference from a global health point of view, especially in some of the less developed countries like India?
Well, that's a very important question, and one that is not easy to answer in a few words. Most of the major advances in medicine had expense initially. Antibiotics did that too. But if they work, they change the natural history of the disease that they target, and something else is going to affect the patient. We all got to remember that when we're born, the deal is we're going to die, and you can't avoid it. So if you look at it from a public health cost point of view, if we take someone with advanced lung cancer who's been a smoker and we cure their lung cancer, they're going to live longer, but they're going to have chronic lung disease, and they're going to be coughing and catch pneumonia, and it's going to get them one way or another. And is advancing life the goal, which I think it is? As we advance life, we have to pay for it. In 1900, the average American lived to be about 45 or 50. Today, the average American lives to be 85. There's a lot of things that happen in your health between age 45 and 85, and we're going to have to pay for it. People complain that 20% of the GDP will probably be involved in medical care. Maybe that's appropriate if it's being spent wisely. I think about a quarter of what's spent now is not spent wisely, and we don't have time to get into all that, and we could be doing a much better job. But the answer, I think the simple answer is these things are expensive to produce. If they work and end a cancer, it's worth the investment. And it takes time to develop these products. And if they don't work, they should be not prescribed. We do not withhold medicine appropriately. It's better done in Europe, for example, where you can't get a hold of some of these new medicines that cost $10,000 each time you are exposed to that medication, unless there's good evidence that it'll help you. So, going back to what was said by one of the other speakers, evidence-based medicine is the answer. In my opinion is, if the evidence shows that it works, let's do it, even if it's expensive, but let's collect more evidence.
Thank you. Do any of the other panelists want to comment on this issue of what is going to be the impact of personalized medicine on global health?
I mean, the one thing, I think we're getting a little bit of the cart before the horse in some ways here. When I've traveled around the world, where Cerner does business in lots of countries, most of them pretty developed healthcare economies. But when I visited Russia a couple of years ago, and as they're trying to recraft the system, one of the longtime folks that was a senior healthcare official in the Soviet era, he grabbed my arm and he said, "Hey, wait a second here. This is all really cool stuff. We need clean water, good nutrition. We got to stop people from drinking. We got to get people back immunized." And I kind of was thinking, I thought, wow, I mean, there is some basics here that still dominate the health needs of the developing world. And I think the best thing we do for personalized medicine is get people clean water, good food, get immunizations, and anti-malarial and all the other public health things that are, we're starting to see impacts on. So I'm a huge believer, and all things are nano, your children's, all things are chemistry. All healthcare is local, and all healthcare is personal. So this is the ultimate personalized business, because at the end of the day, it's all about me. I want to live to I'm 85, and I want to live well and healthfully and all those things. And I hope I don't bankrupt my family or my country in doing it. But that's what I was saying about evidence, the comparative effectiveness. Where do you want to spend your money? Do you want to spend it on getting everybody their MRCT, or do you want to make sure everybody gets immunized, and that's clean water? So I think we still have some tough choices to make, but around the world, I think we start with the basics and build up.
I would just like to add one word, if I may. Thank you. Just speaking quickly around about the misconception that is very prevalent, and that is that the cost of manufacturing a drug is very important for its cost and distribution. The reality is that the pharmaceutical industry is the only industry that I can think of where the profit margins are 90% plus. So the price points are determined by market dynamics. They're not determined as much as some pharmaceutical companies would like to make you believe. They're not determined as much by the cost of research or the cost of development or whatever it is. The vast majority of that goes into profit. What is done with the profit is a different story altogether, but it's verified that the cost of research and cost of manufacturing factor for a small fraction of the overall distribution price. So that is the real question. It's not so much the science, it's about who we are as a society.
Well, thank you very much. This has been a wonderful discussion, and I thank all of our panelists. So please give them a round of applause.